seminar. Today we have a guest from CDC, Anupama Shankar, joining us today to talk about MicrobeTrace. And so she has been with CDC since 2009, working on various aspects of retrovirus surveillance, research, and outbreak response. She has expertise in next-generation sequencing and analysis, as well as phylogenomics. She handles the testing, training, and outreach for MicrobeTrace, and she hopes that you'll find this cool enough to give it a try. So I'll go ahead and turn it over to you.
Thanks so much, Alexandra. Good morning, everyone. Thanks for this opportunity to talk to all of you and share with you a little bit about MicrobeTrace, the tool that we've developed on our team. I really hope you find this useful and try it with data because it has a bunch of different applications. So I'll go ahead and get started. We're going to keep this pretty informal. You can either wait till the end. I'm going to definitely leave time for questions. If you have questions, please do feel free to stop me in the middle. I can pause and answer because sometimes that's just easier. Alexandra, if you don't mind just kind of keeping an eye out for questions or hands raised, and you can just stop me because I may not be able to see that. So, or you can put it in the chat and we can get to it that way. So I'm going to get started now. If you can just let me know if there's any issues if you're not able to see anything. But I'm just going to put into presentation mode. So I'm going to start with a few slides just to tell you about MicrobeTrace, and then we'll go into the live demo. Here we go. So I'm just going to talk to you about what MicrobeTrace does, what the input files are, a little bit about some integration with other tools, and then I'll show you some use cases for various pathogens, and then resources. And please feel free to share the slide deck with everyone so you don't have to take notes as we go along, and you have all of the information in one place to get to whenever you have the time. So whenever you look for a good data viz tool, you're looking for something that's flexible but also powerful, with something with a range of different visualizations, because for different projects or different pathogens, there's different needs. And these should be customizable. Easy to use, of course, without extensive training or expertise or programming skills, things like that. And then that it should be user-driven. So these are the factors we've kept in mind to build MicrobeTrace. So there's a whole bunch of tools out there. A lot of them need specialized skills, and we wanted to avoid that. So all of MicrobeTrace is just point and click interfaces. Very simple to use, very intuitive. It is powerful and flexible. It is completely pathogen agnostic, and it is secure because it uses the web browser on your computer, but it does not upload anything onto a web server. It is a serverless system. So all it's using is the power of Chrome. So what it does is kind of downloads a copy, if you will, of MicrobeTrace onto your computer and then runs it from there. So technically, the first time you navigate to the URL for MicrobeTrace, you can use it once it has downloaded a copy, and then if needed, you can go offline and then continue to use MicrobeTrace. And this is very useful for field applications where internet might be an issue, or in cases where PII, working with sensitive PII, and so you want to work on a secure portal. It is completely free, very important for us in public health. And also there's no need for licenses or software downloads. We all know how that can be. Talking to our IT departments, running through all the red tape, none of that. You just have to navigate to the URL. So essentially, this is just a little glimpse of what MicrobeTrace does. It takes your data, which could be either just molecular, which means some type of sequence data, genomic data. You can also add on demographic data, that's optional. You can add on contact tracing data. You can do all of these at the same time, or just look at one or the other, genomic or epi, by themselves, along with the metadata. It visualizes it in the form of a network. This is just the 2D network. And then we have a bunch of other views as well. Because you're able to combine the sequence and epi data, we find now that, very often the genomic side and the epi side tend to be siloed, and the most enriched analyses will have both of them, and we're just trying to bring MicrobeTrace as one of the tools where you could actually combine the two and give you the whole picture about the outbreak. So these are some of the kinds of questions that MicrobeTrace could help you answer. If you're dealing with disease surveillance or outbreak investigations, you can ask, is this a new introduction of the pathogen, or has it always been in the community and it's just kind of resurfaced? Are there any transmission hotspots? Are two or more outbreaks related? I mean, you might have, and this is very useful for interstate collaboration too. I mean, you might find something in the neighboring state, and if you find that they are related, it could be because of travel between the states. Is there co-infection of two or more pathogens, again, with sexually transmitted diseases, HIV and STDs? This is very relevant. When was this variant introduced and how did it spread over time? Because we have a timeline feature. So we have features incorporated which could help you answer all of these questions about your pathogen. These are some of the linkage types. So for folks unfamiliar with networks, I mean, if you look at a network, suddenly you might think, oh, what is this? It's complicated, but it's really not. It's very simple. All these circles in a network are what we call the nodes, and those could be either persons or viruses, virus sequences, or they could be places. So the node is just a person or a place. The link between them is whether it's essentially a link or an edge, and they could be related by contact tracing. So this person A could have reported contact with B, in which person B, in which case it'll draw a line between the two. If they are related via genetic distance, then it will draw a line if it's below. So you give MicrobeTrace a cutoff value, a threshold cutoff value, and you tell it, hey, anything below a SNP distance of, say, 6, I want you to draw a line if they're related. So, and that genetic distance threshold is pathogen specific. And you need some prior knowledge of the pathogen to be able to say, okay, typically this threshold is what we use for this pathogen to call it part of the same cluster. And so that is another kind of link. You could also have persons reporting exposure, either through a place of work or a place of residence. So it's a person to place link. So just these are the different types of links that you could see in your network. So essentially, these are the types of files that MicrobeTrace can handle. You can have one or more of these. You definitely need a link source file. That link source file can be either molecular, which means they could be either sequences in a FASTA format, or they could be a phylogenetic tree, or a distance matrix, which is essentially distance relationships between those sequences in the form of a matrix, or in the form of an edge list, which is essentially a source column, a target column, which would be sequence A, sequence B, and then the distance on the third column. So you need, if you have molecular data, then you need one of these. If you have epi data, you need a contact tracing file to tell MicrobeTrace essentially what is the link between these people. So this would be the traditional list that EPIs generate. It's person A, person B, who reports contact. And if person A reports contact with 4 different people, you'll have your line list with person A on 4 different rows and then reporting contact with 4 different people. So I'll show you some examples of all these files. This is optional. Metadata is optional, but the richest networks come with rich metadata. I mean, you all know that the final product is only as good as the data you put into it. So if you have rich metadata with as much demographic information as possible about the sequences or the persons, that will really help the analysis. So that all of these will be in CSV or Excel formats. Now we also have interoperability with other tools. So we have the ability to accept JSON files as well as to export in JSON format. And we also have the ability to accept URLs, which I'll show you. So these are the different files. You can use them in whatever combination it is that you have them. This is the era of data modernization, and we try our very best to keep up with the trends, to keep up with the technology. And the keywords now are interoperability and synergy, be it in terms of tools or in terms of departments or branches. The more you work together collaboratively, the better results or the better analysis you have with Outbreak. So in keeping with this, we have modernized our code. We have an Angular version available as a dev environment, in a dev environment. I'll show you that as well. It's just we've improved the backend a lot and made things as easy as possible for the user, and that's available here. Our complete prod version, this one's kind of partial. We have only some of the views. The most popular views are now on this Angular version, but we do have the full prod version as well, functional. We continue to develop complementary bioinformatic tools. As I mentioned, we can accept JSON and URL inputs, and these are the collaborations or integrations that we have already implemented or coming soon. Cedric, which is the platform for foodborne pathogens. If there's foodborne folks on the team, you would know Cedric. We have Ghost, which is actually a CDC-specific platform for analyzing hepatitis sequences. Hepatitis folks, you would know this. Usher is a tool where it builds SARS-CoV-2 trees. It essentially places your query sequences onto into a pre-made database, and then places yours onto that tree. And you can, once you do that, you can visualize that tree in the form of a network just by the click of a button. Same thing with NextTrain. For those of you who are familiar with NextTrain, you can directly view your data. Well, I will qualify that. I'll talk a little bit more in detail about NextTrain. But we have all of these integrations going and more coming along. So I just wanted to talk to you a little bit about MicrobeTrace and NextTrain integration. So this is a powerful tool available for phylogenetic trees. It became extremely popular during the SARS-CoV-2 epidemic because it can process large volumes of raw data, especially for larger genomes. It's very useful. It cleans up, aligns, and analyzes the sequences and gives you these customizable phylogenetic trees. So what you could do is to download, if you are putting your sequences through a NextTrain algorithm, you could download the Genomic files and the TSV files, your metadata, and bring them into MicrobeTrace. Or you can use the integrated button, which is now available. Right now, though, this button on the NextTrain website is available only for sequences uploaded into their database. So if you have your own instance of NextTrain, you would still have to go in and just export your Nuic file and do it that way. But we do have a MicrobeTrace button right here that you could click on, and whatever you're viewing here will be shown as a network in MicrobeTrace. So it's a really cool integration that we have made, and for those of you who are using NextTrain, we would love for you to try this out. This is an example of how the Usher screen looks. This is the SARS-CoV-2 tool that I was talking about. So you can just click this button and view this data, and here you would have put in your query sequence. This is specific to SARS-CoV-2. This is, I just wanted to show you another tool that we have developed in our team. It's called SeqRuler. For those of you who have, a lot of sequences, one thing is that although MicrobeTrace can handle FASTA files, there is a limit to the computational limit to that. And anything above a certain limit, MicrobeTrace will have trouble handling. If it is either very large sequences like SARS-CoV-2 or a large number of sequences, even if you have 1 KB sequences like we typically do with HIV, anything upwards of like 6,000, depending on your computer. If you have a powerful computer, you could probably easily handle 10,000 as well. But anything above that, you're going to start having trouble. And it's not just the number of sequences, actually the number of links, which could be troublesome. So it will be better, rather than using FASTA files, it would be better to generate a distance edge list or a distance matrix outside of MicrobeTrace and then bring it in, because it can handle a lot more that way if you give it a computed distance edge list, so that it does not have to do those calculations. Otherwise, it's going to go in, calculate distances using your FASTA file, and then give you the network. So you eliminate that step by giving it a pre-made distance edge list. And SeqRuler helps you do that. So you can very quickly compute SNP and TN93 distances from your FASTA files. So you have to give it an aligned FASTA file. This is key. Same thing with MicrobeTrace. you have to give it pre-aligned sequences because MicrobeTrace is not essentially an alignment tool. It is capable of aligning HIV sequences because we have built in that capacity because this was originally built for HIV. However, for any other pathogen, we request that you please pre-align your sequences. So this one has a lot of options for handling ambiguities. It is super quick. You can just upload your FASTA files and it will give you the distance edge list in the form of a CSV file. And you can use all your available cores. You can give it the option of using all the cores and it's really quick and you can download really useful free tool that has been developed in our team. So you can download it at the GitHub site there. So having given you a little bit of background, I'm going to actually show you the tool and then we can come back and look at some use cases. So I'm going to exit the presentation mode. Before we begin, let me just show you some examples of excuse me, the files that I'm going to be using. So this is a COVID dummy data set. This is what you call a distance edge list. You have your source sequence, you have your target sequence, which is, or you can call it sequence A, sequence B. You can call it whatever you want. Just when you when you load your sequence into, excuse me, just recovering from a sore throat and cold, please excuse me. So you have your source and your target, and you have the distance here in SNPs. Or if it's TN93, it will be in TN93. If you have any other information about it, you can add that in as well. This one just has source, target, and distance, which is the basic thing you need to build a network. Then we have an epi link list or a contact tracing list. So this is what epidemiologists will be familiar with. You have a source, you have a target, and you have any other information you can about the links. So contact type, for example, exposure, was it high, medium, low? Is the case status confirmed? Anything else you know about the links that could inform your networks. And here, if you notice, I have P1, P2, and P3. These are places. I've just called them P1, P2, P3. And then these are people who have reported contact with that place. In SARS-CoV-2, this was very useful. There were outbreaks in dorms, outbreaks in gyms, recreation centers. So it's very useful if you have a picture of person to place contact. It's very useful to find transmission hotspots. So this is how you would put your information in the CSV file. Now I'll show you a node list, which is where you would store your metadata about these nodes. One thing you have to remember, all of the files that you're using, MicrobeTrace uses this ID column to link them. So these IDs have to match exactly across files, because I know sometimes you pull data through different formats, especially if you're sequencing, if you're getting sequences, you might have local IDs, CSS IDs that come in through LIMS systems, all of that. So just make sure that the IDs match across your node list as well as your distance edge list. And if EPI folks, you're collaborating with EPI folks, then all of these should match. So this is your metadata file. What lineage was it? Collection dates, you could have states, zip code, very, very important, very useful. I'll show you our map feature. You can have anything else you like with it. Profession, viral loads. These are just some examples I put in here. And if you notice, I have a column called node type. This is very useful. Just put it, any patient information, just put the node type as person, and facilities or, hospitals or places, any of that, you can put that as a place node or a facility node. You can call it whatever you like. But if you distinguish this in the CSV file, I'll show you how you can use that information visually to point out places versus persons. So these are the basic types of files. So I'm just going to go ahead and get started with the actual demo using these files. So you navigate to the MicrobeTrace URL, and then you can either drag and drop, or you can use the Add Files button. Just the distance edge list and the metadata file. I'm adding two things. Now the metadata file also has a sequence column, but I do not want to use that. Remember, always use only one source for molecular data. If you have multiple sources, that confuses MicrobeTrace. It doesn't know which one to use. It's kind of put everything together. You'll just get a big hairball. So my source for my molecular data is my distance edge list, and the distance column will give it. So here, this is your settings, very first settings button. You'll see many of these. This is the distance metric you want to use is SNPs, because this is SARS-CoV-2 data. If you're using hepatitis, HIV, any of those, you would use TN93 distances. And we have the default as 16. I'm just going to leave it like that. So this is where you're telling MicrobeTrace that anything related 16 SNPs or less should be called a link. Anything above that threshold, do not draw a link between those two because they are not related. And this is where I mentioned that it's pathogen dependent. So you will put your, and from from helping folks with the SARS-CoV-2 epidemic, from studies, large scale studies that people did, we arrived at the conclusion that anything between 6 and 16 is a good starting point for SARS-CoV-2. Again, it is very difficult to give advice on the exact threshold because it is very dependent on the questions you're trying to ask. Is it a recent or an established outbreak? Are you looking at a small geographical area where things could be a lot more related, or are you looking at a wide area and trying to capture more clusters? So all of those questions will help determine this. So it would be a good idea to start off with some known clusters, play with the settings and see what works best for your question. So that's a little side note about this, how to determine threshold. On our GitHub site, we also have, I'll show you the GitHub site, and we have a little section about suggestions on how to determine starting thresholds for various pathogens. So you just set your distance metric and it's going to build your network for you. You can all your stats at the bottom here. There are 33 nodes in this data set, 46 links based on the threshold that you set, 4 clusters, as you can see, and these are all singletons, which means they are not linked to any other nodes. And it's telling you what your source file was. In this case, there's only one source file for links, which is your genetic distance. You have your main menu up here where you have file. You can add more data. Like I said, if you have epi data to add to this from the same outbreak, you can bring it in. I'll show you how to do that in a bit. You can save. You can open more. You can open a URL instead of using, in this case, I used my files from my computer, but you can also use an open URL. So if you've generated, this is very useful if you've generated an X-strain tree and you have it on your auspice server, you can just bring in that auspice URL and say open URL and then open it up that way. You don't have to download the file and then, bring it in. So that's just a useful feature. If you worked with it for a little while and you want to come back to it, just hit save and it will save it as a session file. So essentially it's a .microbetrace file. You can open it up later. It would have saved all of your changes. In case you want to, you've made some customizations and you want to save it as a style file, it's sort of a template file. You can do that as well using this file option. You can save it as a style file, and I'll show you how to do that in a little bit. That's your file menu. Settings gives you all of your controls. This is you have 3 tabs here. Filtering, which is used only for genetic data. So it's asking you what your threshold is, and here is where you can change your thresholds and see what happens. So I'm changing it to 6, and as you can see, a lot of the links broke off because you're making the threshold more stringent. So now you only have 8 links versus however many you had before. You still have 4 clusters, but they are much smaller. That large cluster fell out. You have a lot more singletons now, 23 of them. So I could just go back and do 16. You can also use this little mini histogram to inform, what it is. So these are, in this range. And then you say, if you click this, it's going to do. So you could just play around with this, like I said, based on the question that you're asking. So we will leave that as 16. I'm going to go to the styling tab. This is where you can color nodes and links by whatever you like. Let's say I'm going to color it by profession. As soon as you do that, you will notice that a key table pops up, which is also customizable. I'm going to leave the color links by. So we could do color links by, let's say, oh, we don't have the link file yet. I'll show you that later on. All right. So this is where I was talking about the style file. So this is where you would apply a style. If you've already saved a style file, you can apply it. So let's say you have data from different states coming in, and it's the same type of data and the same format. So you always want to color nodes by a particular variable and shape them by a particular variable and just save that as a template. So every time you get new data, you can apply that style file, and so all of your data will generate reports in the same format, the same color scheme and same kind of thing. So it's a useful feature to have if you are going to do multiple analyses, same type of analyses with multiple data sets. This is your styling. And then you have your timeline. Your timeline essentially is if you have a date field in your data, I'm going to do collection date, you can see in what order these networks started forming. So if you do that and say done, you will see that there's a timeline that pops up here. These, by the way, are movable. If you right click on it, you can drag and just move this away just so you can see the timeline clearly. And like I mentioned, all of these are customizable. If you don't like the colors, you can change them to suit your needs. You can also change transparency using the slider to go up or down. I've made the purple nodes really transparent if you think they're too much. So this is customizable as well. So timeline, as soon as I hit play, it's going to start playing. As of beginning of July, there weren't any links. And as you go along to the middle of the month, you will suddenly start seeing that there are links forming. And then you can say that there's a whole bunch. So you pause and say, okay, right around the middle of July, there was this cluster that popped up and they were all from the service industry. So, try and analyze that a little bit more. Or if you find that there was a particular strain that popped up at a particular time, that there was perhaps an introduction of that strain during that time frame. So the timeline can sort of help you. I'm just going to move this along. And as you see, somewhere around this time frame, there was a large cluster that popped up here, which had people from various professions. It wasn't really profession related. So again, this is a dummy data set. It may not give you the best sort of story to tell, but you get the drift. If with your data, with real data, it can give you some really useful answers to questions. So that's your timeline. I'm going to take that out. The more features you add, the more crowded it gets. So you can do a lot of things with this. So just see what works best for you without becoming too crowded on the screen. So this was your global various views available to you. This is the 2D network, which is your default. And then there's some other popular views. I'll just show you a couple of them. Before that, let me finish all the main menu options. If you hit help, it's going to open up our wiki site. This one has the introduction to it. You have a whole bunch of topics here. There's like 60 plus pages, all arranged by topic. And here you can go through and just see whatever topic interests you. Or we have full-length manuals. We have tutorial videos. We have a whole bunch of resources, and it's all split out by view as well. And there's also a quick guide video on here, 5-minute quick guide if you don't want to sit through the whole 1 hour. So lots of options on our resource page. And this is also in my slides. So coming back to this, that's your main menu. So on every view that opens, you're going to see these 3 or 4 buttons. This is your settings menu for that view. So this is for the 2D network. You can customize it further. There's 4 tabs in here: nodes, links, network, polygons. Each of them is customizable. You can add labels. You can increase or decrease the label size. You can change the orientation. You can add tooltips, and you can add multiple tooltips. So you say, I want to see the virus name as well, and I also want to see what state it's from. So if you do that, you hover over a node, it's going to show you. Okay, so it's going to show you all of those attributes when you hover over it. So that's tooltips. You can delete this, any extras that you want, which is not being very responsive now. There you go. All right. And then you can change the shapes and sizes. So right now, all of them are just the default shape, but you can shape by, say, node type, right? So node types these are the P1, P2, P3 that I talked about. There are 3 place nodes. And so obviously places this is only a sequence. If you remember, this is just a molecular network. So you're not going to see any connections to places because this is only showing sequence data. But when I add the epi data, you'll see what I mean. So you can shape by place node. You can change the colors, which I already showed you. Same thing with links. You can add tooltips, label. You can size the node by, you can do width by. So you can increase the length of these. So it just kind of separates out the nodes a little bit better. So you can see, especially with larger clusters like this, it's useful to have. And then you can also do width by distance, if you will. And then you will see that, there you go. So you can see thinner lines and thicker lines, and that tells you the relationship between these nodes. So at a glance, you can see which ones are more closely related and which ones are not. We do also have directionality, but we ask that you be very, very cautious while using this feature, unless you're absolutely sure using different data sources that you can determine directionality. We would otherwise be very careful about calling that, especially with pathogens like HIV. It can be a very sensitive issue if you're showing directionality in a network, and we don't encourage that unless it's suited to your needs, in which case we do have that feature. So if you show directionality, in this case it's just a molecular network, but if it's contact tracing and you have someone naming the other person, you will see directionality. So we do have that for you. Network, this is about the network physics. So you can change the charge or gravity. It makes the networks tighter or looser based on the physics of it. And then we have polygons. Before that, I'm going to just take out some of these things. Like I said, it gets crowded really fast, so I won't do width by. I'll just leave it as is. And I'm going to take out the IDs again, and I'll take out the shape. Yeah, we'll just leave some basics. Okay. Okay, so ID, we don't want any labels. Okay, so now look at the polygon tab. If you say show polygons, it's essentially a way to group nodes. By default, it's just going to group by cluster. So this is one cluster, so it's drawing a polygon around it. However, this is customizable. Let's say I want to do it by lineage. So I select by lineage. I'm going to add labels, and I'm going to add colors. Right? So now you have a nice visual about which nodes are part of the same lineage. So if you look at this, you've got all of these, which are B1617. We got a bunch 117s, and then all of these. And then you say, okay, all of these people who are in the service industry who are part of the same cluster are all of this variant. Or if you see a variant being introduced, you can do timeline feature here, and you can see which variant was introduced when. So that's a useful way of looking at this polygon feature combined with the timeline. So these kinds of things you will learn as you play more and more with the tool, depending on what questions you are asking. So that's your polygon feature. These are also customizable, and it tells you how many nodes there are in each of these. It always pops up a key table anytime you do a customization. So these are all that was the settings button. So if you say download, it essentially exports whatever image you see on your screen in various formats, including SVG, which is very useful for publications. High-res image, you can blow it up for posters as big as you like, and nothing will happen to the resolution. So that's your export feature. And depending on what view it is, it will export the file type accordingly. So if it's a table view, it's going to export it as a CSV. But in this case, because it's an image, it's going to export it as a network image. Whatever you see on the screen is going to be exported. This is essentially a center. So if you can zoom in to look at something better, and you say, oh gosh, I zoomed in too much. Ooh, it's a giant network. And so you just hit the center button, it just brings it back to scale. This is called pin nodes. By default, these nodes are not pinned, which means it's governed by the inbuilt physics of the network. It's just going to jump back into its position. If you pin nodes, you can actually drag stuff out. And this is very useful, like I said, for more crowded networks. Some people like to arrange things a certain way and just look at the groups. Another way of doing that, so this is your pin and unpin node. So another way of doing that is to go to settings, go to filter and say minimum cluster size is 2, which means I don't want to see the singletons. So they tend to be hidden. So this is another nice feature. So these were the buttons. Like I said, you're going to see them on each tab. Let me show you a couple of other views. Map view. Essentially, if you have geospatial data in your file, in our file, we have zip code. You can choose any of these, whichever level you have. In our file, we have zip code. So I'm going to select that. And then keep in mind, this is like this blank white map because by default, we've set it to be offline. It's not pulling down map tiles from the map server. But, we can do online, which is what we do for the nice maps. Just say turn on online and turn on, say, show base map. So then now it brings you to this nice detailed map that you're used to. So once you do that, you can see that all of these are represented. You can zoom in as far as you like, and you can see there's a whole bunch of stuff, and it zooms in all the way down to Street View. And so if you click on this, you will see that all of these share the same zip code, right? But then you can see they're still colored by the same attributes that you used before. So everything, all customizations transfer across views. Because you did that in the 2D network, it transfers to map view as well. And so you can see zoom in or out as much as you like. Another popular view is table view. You can see as many variables as you like. You can add or delete variables. If this is not really useful to you, you just delete those and then you add some other things. Let's say I want to see lineage and I want to see profession. So it will show you this. And you see there are 3 tabs here, nodes, links, and clusters. So essentially it shows you all of these in this table, tabular view, and you can download any of these. If you click the download button, it's going to download this as a linked list, source, target, and information about it. So this can be used to further filter subset your data very easily. And this is cluster information. Another view, popular view, is, popular is a very relative term, so I shouldn't use that. So another useful view is aggregation. So essentially you can add, say, state, and then add variable, profession. So if you do this, this is very useful for generating a PDF report. You can see the distribution, the percentage of nodes for each variable. So if you're trying to stratify your data and see what kind of grouping it is, this is very useful to do further statistical analyses. So it gives you this. You can add as many variables as you like and save it as a PDF or a CSV. It has a bunch of options here. So that's your aggregation view. You have epicurve. I will show you our dev version, which has a very nice epicurve. This one's a little basic, but we have improved on it. So feel free to try out the Angular version. I'll show you that in a minute. So let's see, we have dates, collection date. There we go. So you can have cumulative or non-cumulative curves. This is just the traditional epicurve that shows up. And you can click on this and just hide it so it's out of the way. So essentially what you could also do is to drag this. If you see it's open in a new tab, just catch hold of that and then drag it. And then I'm going to bring the epi curve below here. And so you have your sort of a dashboard that you've created yourself. It's very useful for reports and stuff. And then you click this. Well, you will have to center it before you. There you go. Okay, so now you have sort of a dashboard that you can create. So I'm going to close out some of these views and then just show you how to add more data. Here you go. All right, so I'm going to turn off the polygons. We don't need that anymore. And then I'm going to bring in some epi data. So this was just molecular network. So I'm going to say add files and just add. So I'm going to have to go back a little, click through to find it. And here is my epi. contact tracing, right? That's my epi data. I'm going to say update, and there you go. It has added. So if you remember, I turned on the directionality, and as soon as I added this epi data, it started putting arrows in here. So essentially, it's now showing you two types of links. One, which is the dark blue, is coming just from the sequences. So these two are related only based on their sequences. There are light blue, which are related based solely on contact tracing. So that person has been named as a partner. And then there are these dashed lines, which are coming from both, which means that these two people are related not just because they've named each other as partners, but also because their sequences are below the distance cutoff that you gave it. So the dashed lines are actually very strong links because you're coming from both epi and molecular. It's very useful to have that information. Molecular links are much stronger because they're actually showing that those sequences are related. Epi, it could have some recall bias. It could, people may not be giving you accurate information. And so it's good to have these overlaid like this so you can get a much more accurate representation of the outbreak. So these are all the features. I can show you quickly how to save a style file once more to jog your memory. If you save this as a style file, it will save it as .microbetrace again, but you can call it SARS-CoV-2 features and then save it. And then once you go in and open your new data, you can go to the styling tab and say apply style and then browse and then add your style file. So we have a style file here. So you can just add that and it'll apply all these colors and shapes and whatever you did, it'll apply it to your new data. I think I'm done with this. I'll just go back to the slides and show you a few use case examples based on what Jenny told me that a lot of you are working with. I will go back to my presentation and okay. So, all right. So this was a study where they characterized HIV transmission in Australia. This is a very new study which was just presented at a recent conference. They were looking at transmitted drug resistance mutations and used MicrobeTrace for that. I know there's a lot of hepatitis folks in here, so this is where they had used only molecular data. They didn't use epi data, but they have, as you see, they have shaped the nodes by, whether they were positive before or after therapy. They were looking at co-infections, and they are also looking at the number of links. And then they're also colored by diagnosis center, so you can see if there was a specific pattern to that. We have also used it for mpox. When we were our team, a couple of our team members volunteered during the mpox response at the CDC. So we developed. This is a little bit more complex because mpox are very huge sequences. So you have to put them first through NextTrain. So one of my colleagues, Roxy, developed this pipeline where you put sequences through NextTrain, got the output, and then put that output file into MicrobeTrace and sort of looked at how various strains were distributed across the country nationally, and then how clusters formed based on time, spatial as well as temporal analysis. So this is mpox. And then it was also used, Jenny told me that you guys at Virginia's DOH are also working with some crab outbreaks, which I know can be pretty common. So this is where Kansas Health Department used MicrobeTrace to look at some of their crab outbreaks, and they saw a link between facilities and people. And they have a pretty detailed description, which should be in the notes section. I don't want to spend too much time on this, especially I'm always a little nervous to interpret other people's data, but it's in the notes section. So if you guys are interested, just check it out and see how you could use MicrobeTrace to. They have an explanation of this diagram there. This was SARS-CoV-2. Like I said, there was a huge increase in MicrobeTrace use. It was very useful. This was in a skilled nursing facility. And they, again, did sort of a spatial analysis of, where these transmissions were happening. Some resources here for you. We have a whole bunch of, this is our toolkit, if you will, where we have training videos, all of the resources all in one place. This is the actual tool. I will I'll wait for questions and then I'll show you the Angular version because it's pretty cool. It's just a nice looking interface, and we have the full length manuals, publications, whole bunch of things. Can't stop without acknowledging our fantastic team. These are our past alumni. Tony had a huge hand in developing the software in the initial stages, and Els was our activity lead who kind of took MicrobeTrace, was very instrumental in developing MicrobeTrace. And then we have our developer here, Evan and Regan and David. We have our other folks on the team who help with various aspects of MicrobeTrace. We're a small but close-knit team, and we hope that we have developed a great tool and that you can all try it. So I'm going to stop here and take questions. And I cannot see the screen at all. I'm going blind. So Alex, you're going to have to help me. Okay, I see a hand up. Thank you so much for your attention.
Mary, did you have a question?
I was actually just trying to do the applause react. Thank you so much for your presentation. That was great.
Okay, excellent. Thank you. Let's see. Oh, it looks like Bill, my team lead's on the call, and he's been answering questions, so that's great. Okay.
So I think, I guess I don't know if it's a question really as much as a comment, but I think when I've tried to look into using MicrobeTrace in the past, the challenge has been setting up the initial files. So I think being able to export or import like the next strain outputs directly into MicrobeTrace would be a huge time saver and really helpful. So I think that's a really great addition I hadn't seen before.
That's great. That's great. Thank you so much. Yes, you could bring in your Newick file into MicrobeTrace and just add in your metadata. So, so like I said, for NextTrain, we're trying to look into seeing if you have your own instance of NextTrain running on your server, then we would love to be able to have that same functionality where you just click a button on your auspice and then bring it in. But right now that's in progress. So. But in the meanwhile, you can definitely use the features that we currently have.
Yeah. How does it interact with Cedric and Ghost? Because I know you had mentioned those as well, that there was integration happening for those programs.
So for Ghost, you can already do it. Ghost integration has been done for a while. So if you open your Ghost screen, you will see a button that says View in MicrobeTrace. I didn't have that screenshot in my slide deck, I'm sorry, but essentially if you open up wherever you see your Ghost network, there's two options, and one of them should say View in MicrobeTrace, and you can click that button and it'll open up your data in MicrobeTrace already. And for Cedric, it is also a button, looks very similar, but we have had reports from a couple of state users that the screen they see is not this similar screen that we see on the CDC end. So I'm working with our Decipher folks who administer Cedric to see what's going on, and have they really pushed out that functionality to the states, because they told us they were, and that they were going to hold office hours and spread the word to state folks, but that was a few months ago, and just recently I heard from someone in Tennessee that they were not able to see that View in MicrobeTrace button. So for now, what I would suggest is, if you're using Cedric, if you can just export your Newick file, because you can view your phylogenetic tree, correct? In Cedric, if you have the genome module, then you can just download the Newick file and then bring it in that way. Until we figure this out, and I will absolutely give you guys an update as soon as we're able to figure out what exactly is going on with Cedric, because there seem to be, for Cedric, I think there's like divisions of who can see what based on what kind of user you are. So I think it's something to do with that. It's a huge team there with Decipher and Cedric, and I'm trying to figure out who is doing this. Logan?
Hi, thank you so much for this presentation. It was fantastic. So I think I just wanted to clarify that I heard this correctly. It sounds like there is some sort of maybe like limited alignment capability in microchips, but it sounded like it's probably better to do your alignments in like SNP kind of like trees before you actually create your visualizations. Is that correct?
Correct. I'm talking about only about using FASTA files. So if you want to use FASTA files as your input, then they have to be pre-aligned before you bring it into MicrobeTrace for anything other than HIV. HIV is the only pathogen for which we have built-in reference sequences and the ability to align. But for any other pathogen, either you can pre-align outside and just bring in the aligned FASTA file, or you can pre-align, build a tree, and then bring in the Newick tree, or you can just do a distance matrix based on your sequences and just bring in the distance matrix. So either one of these options. MicrobeTrace just needs distance information in one of these formats.
Thank
I
you.
hope that answered. Yeah.
Are there any other questions? You can either unmute or drop them in the chat.
Yes. Yes. So Jenny has put a note in which I will absolutely second. The directionality and arrows can be a bit confusing, and like I said, we just have that feature in because it was requested by some folks. Like we're always looking for feedback, and we have that feature in. But please be very careful when using it. That's what Jenny has put a cautionary note in the chat. And Jenny, do you wanna have your hand up?
Yeah, thank you. Can you hear me okay?
Yes.
Okay, I just wanted to, get your take on that. So thank you for responding to my chat comment. And then also to say, as you're mentioning all the different file types that can be uploaded, and in answering Logan's question, it made me think of another thing in terms of just what you said about the adjusting the threshold limit, whether you're talking about having it use the SNP file or the TN93 distance as the limiting threshold in terms of what's displayed on the screen. I know we have talked a lot about like how it's sometimes confusing for EPIs to interpret the nodes on the two-dimensional space and how they relate to each other. When you make these changes, say to the threshold, or if you pin it and you start to drag the nodes around, it does start to make you sort of question what you're looking at as an epi trying to understand the relationships in two-dimensional space. So I was just wondering if you could comment about that a little bit, simply because, depending on which pathogen you're looking at, it does matter for that that threshold difference, and then also for the different file type, right? Like we try to emphasize in our GenEpi trainings that looking at phylogenetic trees is not the same as a network diagram. And so that's to the point of my comment, right? With network diagrams, you can see, relationships in terms of directionality based on those links, which is very different than looking at the phylogenetic trees. So sorry, that was a bit of a ramble, but I appreciate if you can talk a little bit about our conversations about the 2D space.
Sure. So essentially, I think Jenny was the one who told me to use the magnet, using magnet as a metaphor for, how two things are the links are kind of like magnetic, and they kind of move closer to each other or not. So first of all, the length of the links is not proportional to the relationship between the two nodes. Just because you're closer or further apart does not mean that they're closely related or further related. That's just the physics of the network. So that's the first thing. The second thing is with phylogeny, it's essentially the same thing. It is just showing you relationships, but in a different space. So I will say that this is a note for EPIs. You do need some prior knowledge about the pathogen in order to determine the distance threshold. You cannot randomly change the distance threshold. I mean, you can, but it will change your network in a way that may not be accurate. So what I would suggest doing is, as a starting point, use a priori knowledge. So whatever you would do, if you're looking at a phylogenetic tree, how do you determine clusters? Like how do you determine which ones would be classified as a cluster, yes or no? That's also based on the number of SNPs. So based on a particular pathogen, let's say for Hep C, a TN93 distance of 0.05, according to Yuri, what Yuri told me is what you guys would use to define a cluster. So start with that. And then if you drop the threshold, if you keep making it more and more stringent, you'll see stuff falling out. And those are going to be the most closely related ones, which could indicate that they are part of a more recent outbreak versus established infections. Similarly, if you're trying to figure out, you sort of try to draw a bell curve. So if you keep increasing or becoming less and less stringent, you're going to keep adding clusters, but at some point they're going to cut off. It's not going to go on infinitely. So you have to sometimes, if you have no knowledge about the pathogen at all, then you're going to have to use reference sequences or known subtypes to kind of create that, try that a little bit, find out what threshold works best and gives you discrete clusters. You're going to see the discrete clusters. Let's say if you have a bunch of different subtypes in your analysis and you know that these subtypes have to cluster together. That's what we did for Hantanet. And then you use that, that would be your starting threshold. But for most pathogens, we have some sort of prior knowledge, either from publications or from you guys using it yourself. And like Bill mentioned, I can drop that specific link into the chat right now. On the wiki page, we have a little bit of guidelines based on our interaction with our partners, our collaborators, on thresholds that have been used for various pathogens. I'm going to drop that into the chat in a minute. But I would definitely say you cannot change distance thresholds without a little bit of prior knowledge about the pathogen. And you will have to play with it with some amount of knowledge. Did that address what you were trying to ask, Jenny, or the cautioning?
Yes, it did for sure. And thank you for dropping that link. I was looking for it on the wiki page as you were talking about it. I do think that is a really great addition to your,
yes,
your guidance.
I will absolutely. And I name all these pages different things, and then and then I have to try and figure out. Oh, there you go. Okay, that wasn't too hard. I will drop that in 2 seconds. Okay, here we go. And this will give you a starting point. For some of them, we have the publications. If we don't, then please feel free to drop us a note and we can find out more for you. We are definitely there to help. If you have some sort of questions and you don't really have the time or the resource to try and do all this research yourself, please ping us. We will probably have someone at the CDC, some SMEs that we can ask and kind of get back to you on this distance threshold issue. Exactly. What Bill put in his chat, like I mentioned before, it depends not just on the pathogen, but what question you're asking. For HIV, for example, our default is 0.015, TN93 value. That's what we determine. So essentially anything that's 1.5%, distance should be 1.5% or less to be called a cluster. However, sometimes you will find a lot more in that range, and we want to kind of drill down and find the more recent infection. So we will drop down the threshold to 0.005, which is just even less. So that will give us further insight into within this outbreak which ones are more recent. So you could try doing both and just see how it sorts out. And that's an example for HIV because that's our strong suit.
All right, well, we're right at the hour, so if there are any additional questions, I guess drop them in the chat or come off mute real quick. Is there anything else? Well, thank you so much for joining our seminar this month. This was really helpful. Oh, yeah, if you have time, we can stay on for a minute and look at the Angular version as well, but I know some people might need to drop off.
Yes, let me quickly show this, and if people have to drop off, I understand. I would like to be respectful of the time, but I just wanted to quickly show you. The nice thing with the Angular version is we have an example file built in. So a lot of times people want to try and explore before they bring in their own data. So all you have to do is say continue with sample data set, and it loads this network for you already. This is an HIV dummy data set. It has both epi links as well as genetic links. And then you have all these features. The buttons are a little nicer. The layout is very similar. You've got your main menu, you've got your buttons. Each time you open something, it's going to open it in a new tab. You can, the epi curve is a lot more advanced. You could do multi side by side, and you can do, let's say, where's it? Diagnosis date, right? And then date field 2 is, let's say, infectious period start. So you have 2 date fields, you have different bin sizes, you have tick intervals that you could do. You could change the tick intervals, and then you can do different graph types, you could do overlays. So it's a lot more advanced than our other epi setting. And then we also have a really nice alignment view with a logo and a little mini map, and you could have a lot of features in here too. You can change the layout, sizing, colors, alternative. So we've got some nice features in these new views. Everything else is the same. Table maps all work the same. It's just that's a nicer back end, and then you have these. It's just a little nicer looking. It's easier to close out windows. Oops. You just have to click them rather than do all the drag and drop, things like that. So this is your Angular. It's also, the URL is also in my slide deck. So feel free to try it out. I think I'll just be respectful of people's time and stop there unless we have more questions.
All right. Well, if there aren't any other questions, thanks again so much for presenting today. I've seen your presentations in the past, but it seems like there's been updates since then. So this was really great for me to get a refresher on everything. And hopefully this was new information for some on the call for a tool that we might be able to utilize.
Wonderful. Well, thank you so much for having me, and I appreciate all of your questions and your interest. And it looks like Nicholas had a question. For some of them, unfortunately, we don't have a publication. It's just by talking to SMEs in the field. But if we have them, we will update and add them, Nicholas, for the distance thresholds. Thank you, everyone.
Thank you.